Hi again, and thank you for hanging on with me so far. This is the last of the three-part lectures of this lecture on G protein coupled receptors. And the main topic I want to bring up to you is actually that of orphan G protein coupled receptors. This is an interesting topic that I hope that you can follow with me for quite a few reasons. Essentially, quite a few non-other than G protein coupled receptors are not known to have ligands. That means these are receptors that we know they are G protein coupled receptors because their sequence, their structural similarity to other G protein coupled receptors is clear. However, despite the fact that they are actually found in the genome, and many of them are also expressed, we don't know which is their ligand. Obviously, if you think in historical terms, what has happened, or what happened most of the time, at least in the 20th century, was that the ligands were discovered first, and then the receptors were discovered after. So the discovery of the ligands preceded the discovery of the receptors. With the sequencing of the human genome, there was a paradigm shift, because in that case, when the genome was sequenced, we could identify particular sequences because of their similarity to already known G protein coupled receptors that could be classified as such. But in this case, we had the receptor without the ligand. This actually started the field of reverse pharmacology. So reverse pharmacology refers exactly to that. How do we discover a ligand considering that we know that a receptor is present? And in this case, essentially, we are dealing with a process of de-orphanization. What we want to know is which ligand, what is the ligand to a known receptor. We want to de-orphanize this receptor. We want to actually identify its ligand. And the important thing in this case is, of course, that the ligand of this receptor should have a potency that is actually consistent with a given physiological function. De-orphanization actually has required two types of technologies. One is the purification of active compounds for different tissues. Essentially, we need to find a ligand. We guess that this ligand is expressed in the tissues, but we don't know what it is. So it requires the purification of multiple active compounds. The second aspect, the second type of technology, is to quantify the reactivity of the receptor in heterologous cell lines. What that requires essentially is that with the knowledge of which is the receptor, because we know its sequence, we express this receptor in a heterologous cell line. It's in a cell line that will be expressing this G protein coupled receptor in large amounts. And then we have to expose these heterologous cell lines to specific active compounds and try to recognize some activation, some reactivity of the receptor to different compounds that they are exposed to. Obviously, this can only be done with the existence of high throughput technologies that allow for this monitoring of large amounts of molecules, potential ligands, to the different G protein coupled receptors. Well, you must think, is it so important? Is it so crucial to de-orphanize orphan G protein coupled receptors? Well, there are several answers to that, but the one that actually paved the way to the de-orphanization, the gigantic effort that was placed by the pharmaceutical industries, was of course driven by the interest in finding novel drugs, novel medicines. So de-orphanization when it started already in the in the 90s, at the end of the last century. The orphanization led to the discovery of multiple novel neuropeptides. Many of the G protein coupled receptors that were deorphanized were actually interesting therapeutical targets. And then in this table from a paper by Gavrilesky in 2008, you can find different receptors and the year they were deorphanized and which were their ligands, where were they found, and what therapeutic indication could it have. So obviously, this deorphanization led to the discovery of new medicines, and that was very promising. But you can see essentially that the orphanization, yes, there were some steps, but if the number that I was mentioning in the beginning was already more than 100, well, how many have been deorphanized up to year 2005 and even later years as well? The process of deorphanization was very hyped by the pharmaceutical industry, but eventually it seemed that not all G protein coupled receptors could be deorphanized. There are still many G protein coupled receptors that are orphaned today. Can they be deorphanized? And I think that that calls for a basic reflection on this question. Can it be that a G protein coupled receptor is actually an orphaned receptor? Is it possible that we have G protein coupled receptors that do not have a binding, a ligand, and don't need a ligand? So I think that it would be nice if you can think about this on yourselves. This is just an interesting and relevant question. Why don't you do this exercise for me in a second? Just take 1 minute in which I'm not going to say anything. I'll just keep this slide on and discuss this among yourselves. Discuss this in small groups of two by two, and basically try to come up with a reason why or why not these G protein coupled receptors, or that the G protein coupled receptors may not have a ligand. Take 1 minute. I cannot be with you over there to actually hear what you have to say. But it's an important consideration. It's an important consideration that has been just discussed in the literature. Just another way of looking at this, basically, of the non-odorant receptors in 2013, that's about 6 years ago, 91 were still orphans. And another 37 were still evaluated to verify the orphanization. Of course, the orphan GPCRs varied between the different families. Here you have up to 6 families that are, some of them I presented them, some of them I did not. And then in parentheses, basically, you have the number of orphan receptors, or in this case, in some families you don't have any orphans. In some, you have mostly orphan receptors. In this, the rhodopsin family is the largest one, of course, because you have the olfactory receptors, of which most are actually orphans, but you still have, out of the 284 non-olfactory, 67 are orphans. So there are many, many receptors that are waiting to be deorphanized, if that is actually at all possible. The essential question that you were reflecting on earlier is basically if a GPCR must have a ligand. And I'd say that if you think this broadly, the answer is no. Why should the GPCR have a ligand? A GPCR may be present, but does not necessarily need to have a given function. And this is actually a nice exercise that a researcher called Levoy and co-workers in 2006 did. And he presented in the paper, quite an interesting paper actually, he presented this picture. And this is a hypothetical picture. Notice that in the picture that he presented here, this is a hypothetical situation of things that could be happening with receptors. But what Levoy started thinking is one interesting concept that is well known among G protein-coupled receptors, is that G protein-coupled receptors tend to dimerize in vivo. A dimerization means basically that two units of the same receptor may actually work together. Here you have a dimer, for example. And this would be a homodimer of a non-orphan G protein-coupled receptor. A well-known receptor dimerizes, and in this process, this receptor has a given ligand, can be regulated internally, and can trigger a signaling or an internalization response. What Levoy is proposing is that this dimerization can also take place with an orphan G protein-coupled receptor. In this case, you have the heterodimer formation of a non-orphan in pink and an orphan in blue GPCR. And what he speculates in the article is that the dimerization of a non-orphan with an orphan receptor may result in a change in function. For example, change in function in retention or exportation. So that means the trafficking of the receptor. If the heterodimer is formed, it could hold the membrane receptor longer in the endoplasmic reticulum or shorter. It could affect it actually trafficking. But other possibilities are there, of course, is that it does not affect the ligand is the same, but it may now have different regulatory sequences in the intracellular part of the receptor. So it's no longer responding to the same regulator it was when it's homodimerized, or it could actually react more, or it could actually take on a different regulator. So the signaling and the internalization of this receptor are varied again by being activated or by being inhibited, because the regulator could now be inhibiting. Essentially, if this was the case, why not? The ligand could be different. If the heterodimer is formed, now the ligand that activates this heterodimer could also be different, and that's what Levoy talks about, a distinct pharmacology. And finally, they also recognize the possibility in which the homodimerization of an orphan G protein-coupled receptor could have a constitutive activity. This receptor does not need a ligand, but it will have a constitutive activity in the cell. So this proposition, this speculation that Levoy did now quite a few years ago, actually we have evidence for it nowadays. And this is what I want to just show you in my last few slides in this lecture. This is basically from a paper of Ahmad et al. In 2015 that essentially displays the same situation that Levoy speculated about. What are different opportunities that orphan G protein-coupled receptors offer? And you see basically similar phenomena as I presented earlier. This is the legend of the figure from the Ahmad paper, so I'm not going to say more about this. And here are the two examples that I'm going to basically talk and finish this lecture with. One of the example is GPR50. GPR50 is the mammalian ortholog of MEL1C, which is actually a melatonin receptor found in non-mammalian vertebrates. What happens in mammals is that mammals express both genes, the melatonin 1 gene and the GPR50. And when GPR50 heterodimerizes with MT1, there are things that are happening. So here we have a situation of a heterodimer formed by the melatonin 1 melatonin type 1 receptor, and melatonin is actually a known receptor that binds to the hormone melatonin. But if instead this is heterodimerized, now this heterodimer takes new functions. If actually the GPR50 is heterodimerized with MT1, it's no longer possible for melatonin to bind to the receptor. And this has important consequences. We know from knockout mice in which GPR50 have been knocked out, these mice actually grow less, have a higher metabolism, and show also higher will activity levels. So GPR50, knockout mice for GPR50, an orphan G protein coupled receptor, actually triggers a different phenotype. And this is what you see in these graphs in actometers. These are graphs that show actually activity. The higher the bars, the higher the activity. These are mice during the night. This is the dark period. During the day, you know well that mice are nocturnal. Most of the activity is localized during the night. But notice that there is a big difference between the wild type mice and the knockout GPR50 mice. There is more activity during the night in the GPR50 knockouts than it is in the wild type mice. And then if you count actually how many revolutions or how many turns these wheels that are placed in mice cages do, what you see is that there is an important difference between the knockouts, the dark bars, and the wild type, the open bars. Particularly during the night, this difference is more accentuated during the night. It exists during the day, it's also significant, but it's specifically more apparent during the night. So here we have a situation, knockout of an orphan receptor that we don't know any ligand for, but still through heterodimerization triggers a different phenotype. The same GPR50 is also known to play important roles in thermogenesis and torpor. This is actually a paper published a few years ago about this. And what we know is that GPR knockout mice also readily enter torpor. Torpor means that it's a state in which the animal allows its body temperature to go down to save energy, particularly during the night or particularly in response to fasting. When the animals are fed less, they enter torpor more readily. We know that this response is due to an attenuated response to leptin and a suppressed response to TRH, the thyrotropin releasing hormone. So there is an effect on the hormonal axis in these mice, and this effect presumably is due to the knocking out of GPR50. So GPR50, an orphan receptor without a ligand, still has important physiological functions. And the final example is for GPR20. GPR20 actually works through a completely different way. What we have in this experiment, in these results, for example, is the expression of GRK20. Sorry, this should not be GRK, it should be GPR20. The expression of GPR20 in HEC cells, this is one well-known cell type in which you can express the different components you want, actually lead to a reduction in baseline and also PGE2 stimulated cyclic AMP levels. What happens here? Well, I'll lead you through the data. In the A graph, this is basically a one in which it's just showing that the cells that you can express GPR20 in particular cells. And this is because these are cells that are expressing a fluorescent reporter, and the ones that are actually, so this is actually data from flow cytometry that's showing that you can express GPR20 in cells, and these are the cells that have a fluorescence. And these are the cells that have been inserted with a vector but are not expressing GPR20. And what you are doing is comparing the phenotype, the response between these vector inserted cells with the GPR20 inserted cells. So in this case, what you see is that in the absence of PTX, the cells that are expressing GPR20 show less cyclic AMP. So there is less cyclic AMP, baseline production of cyclic AMP by these cells in the absence of pertussis toxin. PTX is a pertussis toxin. We know well that PTX actually blocks GI proteins. So the inhibitory G proteins are blocked by pertussis toxin. What happens now if we add pertussis? If we add pertussis toxin, we are blocking GIs and the effect disappears. The effect disappears. That tells us that the effect of the expression of GPR20 is mediated through GI. GPR20 actually triggers these cells through the constitutive activation of GI proteins to reduce the level of cyclic AMP. This is also true when these cells are activated with a prostaglandin. So this would show this is the baseline conditions for the cells, and this is when the cells are activated with prostaglandin E2 to trigger a cellular response that is mediated by cyclic AMP. And you see the same thing here in the absence of pertussis and in the presence of pertussis. In the presence of pertussis, no difference, as you also see here. In the absence of pertussis toxin, you see a difference. So the effect in this case, you have a GPR20 that is an orphan GPCR that is actually constitutively activating GI proteins that lead to the reduction of cyclic AMP. No ligand, yet GPR20 has a function. And this is the last example I wanted to do today. Finally, of course, you have examples here in which heterodimerization or the dimerization of GPCR actually shows important pharmacological effects. So this essentially says that heterodimerization is an important function for GPCR, G protein coupled receptors, and the orphan GPCRs could actually have an effect through this heterodimerization. And here you have known examples. This is the end of the full lecture. These are the articles that I have used for the three different parts of this lecture, in particular the ones in boldface. I would recommend because they contain interesting experimental results, and the ones in red are more of a generic review type. Very easy to read in some Nature reviews, in Nature, easy to read and very informative and contain important information about this lecture. Finish with questions. So if you have questions, you can relay them to me via Skype. Let's see if I can answer. Thank you, Francine. Hope these lectures have been interesting, even if it's not the thing that perhaps you are used to. And just fire away, ask as much as you are interested in doing. Thank you very much for listening.